You're welcome back to the fourth episode or session of the 1 million bioinformatician program of Novokin Math Limited, where we will be taking you through the introductory part of genomics. Today, we'll be focusing on the introduction to databases. What are the types of databases that we have? What do they do? What information would you get from these databases? So that is focus in this particular episode. Now, what are biological databases? Going by the name, they are data repositories. They are places in which biological information are deposited regarding the nucleotides, proteins, and the likes. Basically, those two: DNA, RNA, proteins, where they are deposited for usage. So the rapid, and what led to bringing up of these different databases is the rapid rise in the gene and protein identification and its sequencing. So, and it has led to collection of information related to the areas of omics sciences. So this is needed because this effort of understanding the sequence or annotating genes it's a collective global responsibility, so that research can we can have ample information at our fingertips. So biological database is a collection of the biological information that is organized so that it can easily be accessed, managed, and updated. So that is that is the simple definition of what biological databases are. So what kinds of data are stored? You can have a DNA sequence, a full genome, sequence of protein, 3D structures of protein, nucleic acid and protein structure. But basically, just like I said earlier, it is based on the DNA and the protein. Now, there are different types. You have the primary database, we also have secondary database, we also have derived databases. A primary database consists of data derived directly from experiments such as nucleotides, protein, three-dimensional structures of this nucleotide or protein, and they are what we call the primary database. While the secondary database include results of analysis of the primary database and other significant data in form of conserved sequences and the likes. So just by the name, different stage: primary, secondary, derived. Now there are examples of primary database. We have the gene bank, we have the DDBJ, we have the EMBL. So these aspects, we have the ones that are dealing with the protein, we have the ones that are dealing with the nucleotide. The nucleotide database, we have the gene bank, DDBJ, EMBL. So now, not that gene bank doesn't have protein sequence database, but basically you find the nucleotide here, unlike in protein, whereby you will not see the nucleotide sequence database there in the protein part. Now the gene bank is the most popularly used tool database, and it is one of the fastest growing repositories worldwide. It is domiciled in USA at NCBI. I think NCBI full meaning is National Center for Bioinformatics. I've forgotten the I. And it's domiciled in USA in Bethesda. So while EMBL, this is an example of the flight file format of the gene bank. And EMBL, DDBJ is the NCBI, the gene bank part of, is the Japanese part of gene bank NCBI. Because, however, the information between the three, let me wait till we discuss about EMBL. This is how the DDBJ looks like. This is the format of the website. While EMBL is domiciled in the United Kingdom by the European Bioinformatics Institute. So the three have collaboration between each other. So when you have an information is shared between GenBank, DDBJ, and EMBL, and this is the interface of their website. Now, for the protein sequence database, is examples. Is we have the Swiss Spot, PIR, and PRF. So those are the most popular ones. In the secondary database that deals with the protein structure database, we have the PDB, EBIMST, MMDP, and here are structures. We have the secondary database that deals with the domain and motif database. We have those of structure database, gene expression, metabolic pathways, and then we have specialized database. So this is just to give us an information of how this database differ to one another. And here is the derived database explanation. Now, we also have database for various model organisms. So these model organisms, we have many of them. And one of the bacterial model organism, we have the E. coli. So if you want to find out anything about E. coli, you can easily go to E. coli Genome Center that is hosted by the Wisconsin University, and also the E. coli Index by the University of Birmingham in the United Kingdom. For the case of Arabidopsis thaliana, you can search the database for that. That is, whenever you go to all these websites, you would find nothing but everything about Arabidopsis thaliana. In the case of Homo sapiens, Human Genome Resources at NCBI, for Rhiza sativa, for Drosophila melanogaster, Mus musculus, Danuario, C. cerevisiae, and the likes. So here we have Ensemble browsing genome. We have genome browsers. So we also had a session about how to use Ensemble genome browser some while back. You can visit the YouTube channel provided to have an information to know how to use the Ensemble genome browser. And this is how the interface of NCBI looks like. You can visit and search more on there.